Thank you very much, Callum and Mark, for the invitation to speak, and just an honor to participate on this with the great speakers today. So I'm going to speak a little bit about how we take some of this real-world data and evidence and get it regulatory ready, and what we've been working on over at the Smith Center at the Beth Israel. These are my disclosures. This work was funded by a multi-industry group under the governance of the Food and Drug Administration. So U.S. regulators really are challenged with the timely evaluation and identification of adverse events after approved devices in medical therapy. So for instance, IVC filters were on the market for a long time before we really understood that there was risk in keeping these embedded in place with embolization and perforation. More recently, we had a catheter for stroke intervention, which its tip was reliably breaking off during intervention. And it wasn't until some physicians brought awareness to this that the FDA became involved and pulled these off the market. We know this on the medical and other therapeutic side as well, with pharmacies such as Vioxx, where late cardiovascular events emerged after approval. And more recently, we've seen this with the J&J and other vaccines and blood clots. And so really this work we are presenting today is motivated by two aspects. One is there are pre-market randomized trials are not always effective mechanisms to evaluate rare safety signals. And we really, and this is probably the most important part of my talk today, is that we lack a systematic approach to evaluate post-approval safety, in particular for cardiovascular devices. We spend a lot of time relying on volunteer reporting. So we all are familiar with the 21st Century Cures Act at the end of 2016, and this was supposed to create some mechanism by sweeping initiative to incorporate real-world evidence, which is now law, to help address some of these concerns with post-market safety assessments. However, we haven't really seen the realization of a lot of this law to date, and that's an area where I'm going to present where we've tried to help understand how to take real-world evidence and make it regulatory ready to deal with some of these concerns with safety, adverse event reporting. So the case study for my presentation today is paclitaxel-coated devices. And I'm sure most of you, if not 99% of you, are not familiar with these devices, but in the peripheral artery space, we're plagued by really high rates of revascularization and restenosis. And so in 2014 was the first device that was developed that had a drug coating on it to prevent restenosis. This is a picture of a drug-coated balloon here. We also have drug-coated stents. And these made a really large impact in our clinical care. Here it's just target lesion revascularization rates at 2 years for uncoated devices in blue and orange, and now drug-coated devices in gray and yellow. And we can see that target lesion revascularization was almost halved with the creation of these drug-coated devices. However, there was a meta-analysis published in the end of 2018 that suggested there might be long-term harm with exposure to paclitaxel on these devices. Again, these devices were not developed for mortality or survival benefit. They were developed to reduce restenosis. However, these investigators were concerned about some aspect of safety with these, and they did a summary level meta-analysis of published data or presented data at conferences, so not a patient level data. What they found was that 1 year in 28 randomized trials, 4,400 patients, there was no harm with these devices. And this was the typical endpoint for most of these trials for death. Most of these studies didn't go on and look at death after 1 year because that wasn't really part of the effectiveness. It was more of a safety endpoint required for approval. However, at 2 years for 12 trials that reported mortality, they found a marked 3.5% increased absolute risk of harm associated with these devices. Again, going between 1 and 2 years, 3.5% increased risk. And they went out to 4 to 5 years, only 3 randomized trials, 860 patients were involved, but they had a 7% increased risk of mortality. But there are a lot of flaws with these meta-analysis. The trials here were missing up to 38% of survival data up to 5 years. There's no specific cause of death linked to paclitaxel exposure. The more paclitaxel you exposed to didn't mean that you were incurring more harm. Nonetheless, the FDA really came down hard. This is a $250 million industry, and the FDA changed the labeling on these devices, sent letters to healthcare providers, and stated that if these devices do not need to be used, they should not be used. And a lot of institutions, including MGB, and removed them at least temporarily from the shelf. Now, normally we respond to safety concerns like this with potentially a randomized trial. And we saw this happen for stent thrombosis rates in the late 2000s and what led to the DAP study, looking at extended duration dual antiplatelet therapy. However, a concern here was really the sample size estimates needed to power a study to find a signal of harm at 4 to 5 years. And so, again, these devices are class 1 in guidelines. They were being regularly used. And if you wanted to power a randomized trial to 3 years, you would need more than 40,000 patients, and to 5 years, more than 20,000 patients. And this would be far and away the largest randomized device trial for peripheral vascular intervention ever. So we became involved at this point because the FDA was looking for an opportunity to really generate regulatory level safety evidence, but from observational data. And so we've kind of created a template. We now are involved in several studies looking at safety assessments for peripheral and cardiovascular devices about how we went about this. The first biggest question, obviously, is choosing the right data source. And we've heard a little bit today about all the different areas where we can be extracting big data, real world data, whether it's from EHRs, from social media, from data sets like billing data sets. And we really settled on here the Medicare database for several reasons. For patients who are diagnosed with peripheral artery disease, 1 in 6 of them are over the age of 60, which means they're well replicated, well represented in the Medicare database. There's high ascertainment of mortality in this data set. And very importantly, there's specific codes. And I'm sorry this is busy, but just to show you, there are specific codes for the type of device that's being used, for the specific artery that's being treated. And we actually went through over 1,000 procedures to validate these codes, and they had incredibly high negative and positive predictive values. More importantly is if you want to get reimbursed for using these devices, you have to use the appropriate code. And whenever reimbursement's tied to coding, misclassification falls dramatically. We also really spent a lot of time on experimental design with feedback from the FDA. So there's certain requirements from the FDA to make data really regulatory ready. One aspect of this was settling on a non-inferiority design, as these devices were not meant to improve survival, but really be no difference in survival between an uncoded and a coded drug-coated device. So we worked on the non-inferiority design and negotiated a margin that was appropriate for both sides. We also had to create data sets using a propensity score-based method called inverse probability of treatment weighting. And this was supposed to be separate from the outcome. So we make a data set that we weight the patients so that they appear similar and then connect it to the outcome data set so that there really is fidelity that we are not manipulating the data. So a lot of trust also that goes into making regulatory ready data. We also held ourselves to the same standard as many people on this conference call are trialists, that we were transparent, that we had a pre-specified design that we followed to the T. And this was a paper that we published, a methods paper on circulation, cardiovascular quality and outcomes that really outlined exactly how we were going to approach this regulatory question. This is also publicly reported on the clinicaltrials.gov website. But I think the most exciting part and unique part about this analysis is really how you draw causal inference from observational data. And in particular, when you're doing comparative studies with devices. And this cartoon here just demonstrates some of the issues with non-randomized data. And I like to give an example of really what plagues the most of these studies is treatment selection bias. So, this was a well-known study. There's several of these looking at coronary stent and survival in the late 2000s. And what was of interest was whether drug-eluting stents had a survival benefit over bare metal stents. Now, I'll prompt you that in randomized trial data, this was never seen. But we saw a number of real-world studies using methods such as propensity score matching and stuff many of you have heard of to try to evaluate survival. And time again, they were finding that bare metal stents had worse survival than drug-eluting stents. But there's some concerns with this, particularly because we haven't seen this in randomized trial data. But we also see, that there's some impracticalities of the data, such as survival starts to change pretty rapidly after discharge. You can see some separation of the curves here. And you wouldn't expect the benefit of a drug-eluting stent to occur for much later, especially because most of its benefit is through decreased target lesion revascularization and decreased need for more procedures. And so we all know from our own clinical acumen that when we were using bare metal stents, we were doing that for a reason. Most of these patients were either, had an upcoming surgery, maybe had a GI bleed, were frail, had poor adherence. And it's really hard to take those two populations and make them totally similar, especially when a lot of these variables are not available in claims databases or registry databases, and sometimes even in our EHRs. So the biggest aspect of regulatory science is making sure that we find a risk estimate that we feel confident in. And by doing that, we really want to pressure test our statistics. So we designed this study with 3 different approaches to inference for our causal analyses here. This first one is instrumental variable analysis. falsification endpoints, and a missing confounder analysis. And for the sake of time, I'm going to give you just the tip of the iceberg here, but these are pretty unique approaches to causal inference. The first here is an instrumental variable analysis. And again, we always are trying to find an association between a treatment and an outcome. And we're trying to adjust away these measured confounders, either through multivariable regression, propensity score analysis, and whatnot. But we don't always know, we don't always have control over these confounders that influence a decision to treat someone or not. And so the instrumental variable allows us to leverage some of the randomness that occurs in the world to assign treatment. So instead of the treatment assignment being based only on the patient's characteristics and the decision of the operator, some of these treatments we know occur just based on the time of the year, the place, the location, and other natural aspects of our environment. And by doing so, we can reduce the risk of stuff such as treatment selection bias. So for an example, in this analysis, we looked at the use of drug-coated devices. This is a histogram showing on the x-axis the proportion of drug-coated devices that are used at an average hospital. And then the y-axis is the percentage of those hospitals. And what we found is incredible variability. There's some hospitals that don't use a lot of drug-coated devices. Some of this is due to costs of these are more expensive than uncoated devices. Some of this is due to the fact that they don't believe maybe in the data. On the other hand, there are centers that use a lot of drug-coated devices, and they do believe in it or are willing to pay for it. But the bottom line here is that these decisions are not necessarily being made because one hospital is treating more sick people than another hospital. It's due to other decisions that are being made at a higher level than the patient. And so what we look at is really if that same patient walked in a hospital down who don't use these drug-coated devices and another hospital that does use them, they're going to get exactly what that hospital does. It doesn't matter what type of patient they are. They're not going to get a drug-coated device if they go to this institution, and they are if they go over into the high users. And so that is really the basis for an instrumental variable analysis for this drug-coated device question is, what is the variation in use among hospitals, and how does that influence all-cause mortality? Another really important aspect here, and for many of the scientists on the call, is something we like to call a negative control experiment or a falsification endpoint. And an example I'll give here is if we ask the question is whether a seatbelt user has a lower risk of car-related death. Okay, so our question here is we want to look at people who use seatbelts and don't use seatbelts and see if that seatbelt reduces the risk of a death related to a car accident. The concern, however, though, is that maybe we could account for factors such as education, income, where they live, but there might be behaviors that we can't control for, such as people who use seatbelts are more likely to be rule abiding or more likely to follow the speed limit and other aspects there. So we want to make sure there's no differences in these two populations when we control them. So we look at an endpoint that's not related to the exposure or seatbelts, but something totally unrelated, but makes sense in this pathway. So here we look at a falsification endpoint of paying your taxes on time. So potentially those seatbelt users who are more rule abiding are going to pay their taxes more often than the others, and this can influence the ability to balance those patients at baseline. If this endpoint, which is not related to seat points, remains positive or associated, we know we have some issues between the two populations that are uncontrolled. So for our analysis here, we looked at drug-coated balloons in 3 endpoints: heart attack, congestive heart failure, pneumonia. And we didn't see any relationship between that drug-coated balloon and any of these endpoints. And we want to imagine there'd be a relationship here because drug-coated balloons do not reduce the risk of any of these adverse events. Lastly, we want to pressure test our analysis to make sure that it's really resistant to the influence of uncontrolled confounders. So we look at these unmeasured confounders, they're really a dummy variable that represents something that we didn't control for. And we look at it in different ways. We look at the prevalence in the data set and how prevalent it needs to be to overturn the results. We look at the association with treatment, so how associated it might be with a drug-coated device, like a typical confounder. And then we also look at the association with the outcome, or in this case, death. And we look at different measures or strengths of this. Now, this is a unique plot. It's a little complex to look at, but what we're trying to find is variables that fall in this upper left-hand corner or variables that fall in the right-hand corner at different prevalence levels to see if we can overturn our results. If our findings are robust to these uncontrolled confounders, then we feel very confident that even if we are missing some variables, it's unlikely to change what we were detecting or trying to detect in this analysis. So I'm going to show you some preliminary results of these work. This is going to be presented as a late-breaking clinical presentation at the ACC in a few weeks. I'm only going to give you the preliminary results here that we presented to the FDA in 2019. So we looked at 150,000 patients treated between 2015 and 2017. And we found here in red, there is no associated between drug-coated devices, and in blue, non-drug-coated devices. So this was a propensity score-based analysis, traditional analysis that I talked about earlier. We found no evidence of harm. As importantly, though, when we looked at instrumental variable analysis, we were able to replicate those same risk estimates by using a really orthogonal approach here. Again, we looked at variation in drug-coated use by institutions, but we got a very similar risk estimate of safety associated with these devices, all of these really close to that 0.95 we found in the propensity score analysis. When we looked at those falsification endpoints, which we'd expect not to be influenced by drug-coated devices, for a heart attack, congestive heart failure, pneumonia, there was no association. So really overlapping curves between those who got drug and non-drug to suggest that our patients were pretty similar after we did adjustment, that there weren't any residual confounding concerns between our patient groups. And lastly, for that hypothetical or dummy confounder, we found here that if we were missing any of our measured variables in our analysis, so let's think about age, diabetes, chronic kidney disease, even if we were missing those, none of them would have been at that strength in this upper left-hand corner, the lower right-hand corner, to overturn our results. So really, we feel that our findings are robust to uncontrolled confounding for that reason. So as I mentioned, we're going to be presenting these at the ACC in just a few weeks. This is really meant to be a template of how we've been approaching these regulatory decisions and safety concerns for the FDA. And we have another study looking at this in the same kind of systematic approach we've been going through. I want to thank everybody who's been working with us over at the Smith Center and our entire vascular research group. And I'm going to stop here to allow for any questions. Thank you very much again.
Eric, thank you very much for a great talk. We have time probably for one or two questions. I'm going to take the liberty of asking you myself, have you looked at a time domain in all of this? It strikes me that some of the benefits from this would only be further emphasized if you were looking at data at different times in the deployment, let's say, of a new intervention like a drug-eluting stent. Is that something that's helpful, or is it just, do you end up with problems with sparseness of data versus dense data at different times in the analysis?
No, that's a fantastic question. That's actually on the docket for stuff we're working on in the future. But that, we look a lot of these things in this analytic methods called difference and difference or time interrupted series. And so going back and saying, what does mortality look like before these devices came on the market? What happened when they come on the market? And then what happens after they get pulled again is a great way to look at it. There are always environmental or external concerns there, or other practices changing during the same duration of time where maybe we're better operators over an 8-year period. And also after these devices got pulled, there's some concerns that higher risk patients were being treated with the few devices that remain. But this is a very insightful question and something that we've been thinking a lot about, and you'll be hearing more about it.